Hi, I'm Tommaso, PhD at Graz University of Technology.
And I'm Daniel, also a PhD at the Graz University of Technology.
Welcome to Bridge the Gap, the physics-oriented podcast,
in which we give insights into various research topics and the life of scientists.
And by the way, if you want to contact us, you can drop a line to our email address bridgethegap@tugraz.at. Hey Daniel.
Hey Tommaso.
Do you know about Eliza?
Not really.
And what about Simoa?
Are they friends of yours?
Hey, and here I thought you were a bit more on the sociable side than I am.
Well, who isn't?
Well, Eliza, Simoa, they all sound like female names in Italian.
Okay, so either I lost it or you did, because I have no idea what you're talking about.
True. Sorry for letting you down. As it happens, though, those are the names of two experimental techniques used to detect biomarkers potentially signaling neurodegenerative diseases.
So we asked some students and colleagues of ours what they knew about neurodegenerative diseases in general.
And that is what they had to say.
These diseases cause the degeneration of brain functions by destroying brain cells, which causes trouble with memory, movement, speech, and other factors. An example would be Alzheimer's or Parkinson's disease. And although these diseases are so prominent in our world, we barely know what causes them.
Neurodegenerative diseases are diseases like Alzheimer's or Parkinson's that attack the brain, which means that the patient will slowly lose their mental and physical capabilities. And as far as I know, not much is really known about such diseases yet, so there's still much research to be done.
What are neurodegenerative diseases? I think they're not good to have, and I don't want to have one, for sure. And how much do we know about them? Honestly,
nothing.
Basically nothing. Not enough to, heal them or something.
It's something, I guess, in the brain, which at first it's maybe not that present, and when it goes on, it's getting worse and worse.
The neurodegenerative diseases should be the non-reversible illnesses or damages done to the neurological system. And I'm not an expert, and I would guess that we know some things about them, but mostly not so much on why exactly they appear, why do they develop, and why some individuals are more prone and the others less.
So, today with us we have Dr. Rina Demjaha from Medical University in Graz. Rina, stage is yours. Could you please introduce yourself, your background, field of expertise, please?
So, hi guys. First of all, thank you for your kind invitation on this really nice podcast that I'm a big fan of. So it's a big privilege to be here finally myself. As you said, I'm Rina Demjaha. I'm a medical doctor, currently doing my PhD at the Medical University of Graz at the Department of Neurology, and my main focus is on biomarkers on neurodegenerative diseases.
So could you tell us a bit more about neurodegenerative diseases?
Yes, of course. So neurodegenerative diseases are basically neurological conditions that are characterized with loss of function of the nerve cells that we call neurons. And they're very, very complex diseases. Therefore, their management is also super complex.
So what makes it so complex? Why is it so hard?
Well, first of all, the beginning of these diseases is usually very silent. And actually, by the time that the patients come to the clinic with the symptoms, there's significant damage already done. Besides this, as I previously said, so they're very complex and the mechanisms are not very well understood to this date. And even though there's a lot of research going on, there's still not very clear things on how the mechanism is, and this makes it also very hard for the treatment. So that's why most of these diseases basically have a symptomatic treatment, which means that doctors treat the symptoms and make the disease slower, but they cannot heal it fully.
So, you mentioned neurons. Can you give us a brief introduction about what a neuron is, and so we can go through?
Yes, of course. So a neuron, as I said, is the main cell of the nervous system, and it's consisted of three main parts. So that's the body of the neuron, the dendrites, and the axon. The dendrites receive the neurotransmitters, which are these substances that basically send the impulse through the axon to the other neuron, and that's how the whole signals in the brain are sent from one part to the other.
And so when we have these neurodegenerative diseases, what happens is there's a some damage occurring in the central part or the extrema? Like, what are they characterized by?
So there are many different neurodegenerative diseases. Some of them, for example, you might have already heard of, like Alzheimer, Parkinson, multiple sclerosis, and they all have different mechanism. If I talk about MS, which is basically the disease that I'm mostly focused on, is an immune-mediated disease that is consistent of basically inflammation and neurodegeneration. And it also has different types of this disease. So it can be relapsing, remitting, but also progressive. So what happens is that the body itself creates an inflammation, which then harms these neurons. And the axon basically is damaged. So the information is not very well transmitted from one axon to the other, from one neuron to the other. And this then causes problems. Overall, these neurodegenerative diseases have different symptoms. For example, they can have problems with cognition, movement, sensation, but also with emotions, etc.
So now, what is the usage of biomarkers in this context to diagnose or maybe help patients?
Biomarkers have been used in medicine for a long, long time. You might have heard of, for example, troponin, which is used for cardiac infarction mainly, or different hormones to check the different organs. But in neurodegenerative diseases, the main problem was that the biomarkers that were available were not measurable in the blood. They were mainly measurable in the cerebrospinal fluid. And in order to obtain it, it's a very invasive procedure. But with the development of the technology, now we have assays that have made it possible to do the measurements also in the blood. And this has opened new era for the neurology and neuroscience overall to make measurements in the biomarkers. So what are biomarkers actually and their meaning in neurology? I can say that, for example, MRI scans are also markers, but they're imaging markers. And in order to see differences in the MRI scans, we need a longer time. However, with the biomarkers, it's possible to actually detect the changes more earlier than on the other imaging markers. That's why they have opened a new door to the management of the disease. They also can help us with the treatment, the disease progression, the prediction of relapses in this case, for example, in multiple sclerosis and so on.
So what biomarkers are there that you are looking at? And also, how do they come into the bloodstream? So are they parts of the neuron, or what are they exactly in this case?
So basically, while this neurodegenerative disease develops, there's a constant inflammation. And during this inflammation, the substances get secreted to the cerebrospinal fluid and then to the blood. And as I said, the concentration is much higher in the cerebrospinal fluid, but then also they somehow escape the system, and while getting filtered out, they also get through the blood. There are many different biomarkers available, depending also on the disease. So there's biomarkers for treatment response, for progression, for prediction, also to check the pharmacodynamics. So depending on what you want, you can check the biomarkers. So the two main biomarkers of neurodegeneration are NFL and GFAP, which are also the two most promising clinical biomarkers.
Can you tell us about these two main biomarkers?
Yes, of course. So NFL is a part of the axon. So when there's a neurodegenerative disease or inflammation happening or whatsoever damage to the neuron, it gets secreted to the CSF and the blood. Whereas the GFAP is an astrocytic biomarker, which basically gets secreted from the astrocytes. And astrocytes are actually these star-like cells that also create better biochemical environment for the neurons. They also provide nutrients to the neurons, and also, for example, they remove the toxins, and they have a big role at the blood-brain barrier.
I assume, since you say both are important biomarkers, that different information can be derived from them. Can you elaborate on this or explain to us?
So, as I previously mentioned, there are different types of biomarkers, right? So there are some that are very disease-specific, like phosphorylated tau protein, for example, which is specific for Alzheimer. However, in neurology, most of the biomarkers are actually not disease-specific. Most of them are, however, very sensitive for neurodegeneration. And there's a lot of studies going on, which actually now suggest more of a combination of these markers for a higher sensitivity. GFAP has shown a great potential when it comes to predicting the clinical worsening, especially in MS patients, whereas NFL is better when it comes to, for example, acute causes.
So say now you run a blood test and you find a given concentration of this or that biomarker. Is there a unique one-to-one correspondence with the disease? You said probably this is not very often the case. So what do you do after having checked maybe the blood of a patient and something? How does the procedure work?
That's actually a very nice question, and it's something that we get asked a lot in the clinics because I have the feeling that people have always this thought that medicine is black and white. And actually, medicine overall is a gray area. It's very important to always keep in mind that there's a lot of factors that come together before you come to a conclusion. So with that being said, we always take a full look on the patient. So we need to know the clinical examination. We check also at the imaging, and with the biomarkers and everything else combined, then we can reach to a conclusion about the disease or the overall progression, the treatment response, and so on.
So is this already used clinically, or is it still more on a research level?
NFL is actually in one foot in the clinics and in one foot in the research, while most of the other biomarkers are still more research-based. However, there are countries in the US, especially, that are integrating this in the clinics. We also, in Austria, we are working towards that direction, but there's still a lot of work going on.
So how exactly do you use this? What do you tell a patient when they have a specific level of something? What can you advise them, or how can you help them with that?
So what is very important to know that these biomarkers have also confounding factors. So we have to first correct for the confounding factors before we use them. And we usually use the Z-score. So there's not a specific cutoff level for these biomarkers yet. However, we use it for ourselves in the clinic. So at this point, it's not something that reaches the patient directly because we use it for our own orientation when it comes to changing the therapy. Let's say a new patient with multiple sclerosis has started a treatment, and then you do the blood measurement, and then you see that the neurofilaments are still higher than they should be for a person that doesn't have a relapse. That would make us think that this therapy is not working very well on this patient. Therefore, we would start thinking of changing the therapy.
So since you're a medical doctor, I'm really fascinated by the measurement methods, because for us, everything is theory, or simulations. So what exactly is that you do during your day if you have to measure something? Are there some measurement techniques you'd like to talk about in here?
Well, yes, because I've been talking all this time how the new doors have been opened and how now it's possible to do these measurements in the blood, and this was not previously possible. The method that we actually use, and we're very proud to be the first center in Austria who has this machine, is actually the SIMOA machine, which stands for Single Molecule Assessment. SIMOA is basically bead-based ELISA technique, which uses these magnetic beads in order to do single molecule assessments. And this has made it possible that once the measurements are done in just one molecule of this immunocomplex, there's a much higher sensitivity, and it's easier to detect the filaments or other biomarkers also in very low concentrations. So that's how basically we use them in just a normal blood collection in the serum.
So then what is ELISA actually, since you mentioned this?
ELISA is one of the most famous ways to measure proteins, but not only. There's different types of ELISAs. There's direct, indirect, sandwich ELISA, but there's also other methods that are used in the labs for such measurements. Also, like Western blot is one of the methods. But as I said, we basically use the most recent and this super highly sensitive machine, which is actually 1,000 times more sensitive than the normal ELISA.
Since you mentioned before that there can be several confounding factors, I wanted to ask, do you do statistics on these samples? So you need to measure your blood samples or the concentration of some biomarker in the blood sample several times to be sure, or is it a one-time process? How does it work?
So yeah, the SIMOA machine actually is very super well automated and is fully automated. So if there's a problem that happens with the measurement whatsoever, which is not very common with a fully automated machine especially, it usually tells you why there was a problem. Like, for example, if there's a lot of fat in this sample, that can make the measurement not so possible. That's why we, for example, centrifuge the sample and we try to omit that part that has the fat. But when it comes to the co-founding factors, we always, before interpreting the results, we use a Z-score, as I said. For NFL, they're corrected for age and BMI, whereas for GFAP, we also have to correct for sex, because research has shown that females actually have higher level of GFAP, also in normal aging people.
Since you said there are differences based on sex, are there also differences based on the age, on the physical activity someone does? Does this influence the levels of these biomarkers?
Yeah. So the main co-founding factor, especially for NFL, is age, especially when it comes to people that are older than 60 years of age. Whereas in people younger than 60 is mainly the body mass index. However, NFL is also affected by stress or smoking or also pregnancy, for example, and many, many other factors.
How do you make sure that the CIMOA counts the correct proteins? Since I would intuitively say, but correct me if I'm wrong, that there exist proteins that have similar shapes also.
So since CIMOA is based on this ELISA technique, what it does is that it has target proteins that we before know. And according to that, we also get the kits, and that's how we know which proteins are being measured. So we don't just do a measurement and see which proteins come up. We have targeted proteins, and then that's how we know which protein we're measuring.
It's like playing Lego.
Yes, pretty much like that.
So you said before your main focus is on multiple sclerosis, right? Is it understood why this happens? Meaning, like this neurodegenerative process that happens, do we know the causes, and is there something that one can do to prevent it, for example?
Well, prevention would be a good point, and is something that researchers and doctors are really working on. As I said, the mechanism is not very well known. However, it is believed that Epstein-Barr virus can also trigger the immune system to actually get overactivated and then start
targeting ourselves.
Targeting, exactly. But Epstein-Barr virus is a very common virus, and there's a lot of people that have been infected with it and don't have multiple sclerosis. So there's also environmental factors and also genetic factors that play a big role.
I understand what you mean by gray areas.
So do I understand correctly that it's an autoimmune disease, sort of?
It is an immune-mediated disease, yes, exactly, where the body itself basically attacks on this neuron and the myelin sheath.
Is this always the case for neurodegenerative diseases?
Not for all of them. So as I said, they all have different pathomechanisms. So they're not all autoimmune diseases. This is mainly for MS, but they all have a different mechanism which is specific for every disease.
Since you do research and clinical work now in parallel, would you say you benefit from this?
I think research is crucial when it comes to medicine itself. I think it's a big part because when you're a medical doctor, you basically see the end part. You see the patient, you see their symptoms, and you try to treat them. But actually, there's a big work and ongoing research that happens so that you are able to give those medications or even the steps that you go towards diagnosing something. So I think research is a very, very important part. In addition to that, it's very good because somehow it gives you hope and it motivates you to go even deeper into research and find out these unsolved pieces of puzzles that we still don't have especially in neurodegenerative diseases.
So you also said before that these diseases cannot really be treated and there's more like a symptom treatment sort of, yeah? What do you think, when will we be able to find solutions for this, like that fully heal people?
Whoa. I think if I would be able to give the answer to this, I would really get a Nobel Prize tomorrow. However, this part is not as gray as I said that medicine is. There's a lot of promising steps. There's a lot of treatments that have shown good results, and we're very, very optimistic when it comes to this. The exact timing I cannot say, unfortunately, but I think it's in the near future that we really will be able to have even better response to the treatment that we give to these patients.
So you think it's also possible to go towards a patient tailored cure or something in that direction once we're able to understand what triggers the disease or how the patients react and something, so also something in that direction?
Yes, exactly. This is not just in neurology. That's what's happening in today's medicine. So there's patient and there's disease. And we should always treat the patient, not only the disease. Until now, people and medical doctors have been always taught to treat the disease. But now we're going more towards treating the patient, because every patient has a different reaction to maybe the treatment of the same disease. Therefore, this patient-oriented treatment is very common thing that's happening now, and also neurology is going towards that direction. And biomarkers are actually helping a lot in this.
I think you hinted this aspect, but I'll ask the question anyway. So, which is the aspect about your research and your work that you enjoy the most? Because you seemed pretty excited about the fact of being able to do research and also being a doctor at the same time.
It really fulfills me. I'm one of these people that really enjoys their job, even though it takes a lot of time and free time and energy. I just feel like being a medical doctor, you have the opportunity to touch into people's life and make it better most of the time. Unfortunately, that's not always the case, but at least you do all you can to make them feel better, to repair their impairment when it comes to movement or like cognition and all these kind of different diseases that we have. And I think it's really an honor and a pleasure to be able to treat such patients.
Maybe also a bit more personal question, because you're from Kosovo and then you went to Turkey to do your medical degree. Then you came to Austria to basically redo many exams in German in parallel to doing your PhD. How do you feel about that? And was it hard, your journey? And how do you like it here also?
So when I went to Istanbul, it was a totally new environment for me, and I was only 18 when I moved out. Learning Turkish was also quite a challenge, but it was beautiful because I really enjoy learning new languages, and I'm a very goal-oriented person, so I knew that I went there. I left my family, my comfort zone for a specific goal, which was to get a good medical education and become a good doctor. So that's why I actually studied a lot, also to learn the language, but also to finish the university. And then I also wanted to come to Austria for the PhD because, unfortunately, Turkey had some still has problems, and research was not very well supported. And then I had to learn, of course, German as well to nostrify my diploma, which also included like almost half of my medical studies to be repeated. It was hard. German is a harder language than Turkish, I have to say. But as I said, I truly enjoy what I do, and I enjoyed relearning everything in German as well. And the thing with medicine is that it never actually ends. You know, even when you're graduated, there's new treatments that are being introduced. There's new diseases sometimes as well. So it constantly changes and you have to be updated anyways. So for me, it was just a continuation. It was not hard, but I think it was not easy. But I think when you have something in mind, you always can do it.
So if listeners remembered only one thing of this podcast, what would that be?
I really hope they remember it's important to really go to the medical doctor as soon as you feel something, as soon as you have symptoms, and actually also get regularly checked up. I hope they remember the importance of biomarkers and also believe in science. Because I have the feeling that there's still a lot of people who don't really believe in science and scientists. And I really, really want to tell everybody that our main goal is to actually bring better things to the clinics and science. So they should believe in science.
Well, on this note,
Very nice.
thank you. Thanks for being our guest, and see you next time.
Thank you guys for having me.
Thanks for listening.
And hear you next time.
And by the way,
if you want to contact us, just send a mail to bridgethegap@tugraz.at.